Today I'm going to be talking about functional and structural imaging of a cortical circuit. But before I unpack that, I want to say that I've at least a third followed the advice for the talk, talking about future directions and big unsolved problems. But the first third of the talk will be a discussion of a very broad overview of the kind of work that's being done in my lab right now and the nature of the observatories that I hope to bring to the Allen Institute. The middle part is posing a question, and the last part is looking at our first attempts of applying certain techniques to answer similar questions and reposing the big question at the end. And the big question is the interaction of two separate questions. The first is, what does a circuit do? And that is, at least in my book, answered with in vivo physiology, seeing what a circuit does or individual neurons do in vivo during a task or during sensation. And then a separate question, how is it wired up? How is exactly the same circuit wired up? And most importantly, what's the relationship at the single neuron level between neuronal function and interneuronal connections? So two separate kinds of data. First, the physiology, and the current physiology that's going on in my lab is mostly two-photon calcium imaging, where we can make pictures like, as you see on the left, where this is a picture of roughly 100 neurons, and where we study the physiology of these neurons. I'll show you what the actual physiology looks like later, but the output of these physiological experiments are functional labels, such as the canonical thing that one studies in visual cortex. All of my work, I've really been essentially working on this question since I've had my own lab 15, almost 20 years ago, looking at the relationship between vision and connections. I used to do that kind of work with single electrodes, looking at the receptive field of neurons, and with single electrodes looking at the correlated firing of neurons and inferring the connectivity. Now we're actually doing the physiology with optical techniques and the anatomy with true anatomy, I'll tell you about that later. So the physiological techniques are calcium imaging, where we can color code these neurons according to the orientation of the stimulus that excites them. So here is a physiological experiment in the mouse, where we have roughly 4 different colors, and the colors correspond to the orientation preference of neurons. The red neurons prefer horizontal stimuli or respond to horizontal stimuli. The green neurons respond to vertical stimuli. On the right is an anatomical image, and I'll tell you how we got it in the last third of the talk. And we have 10 neurons here, color-coded, the same color code, and a bit of the network that those neurons participate in. So very quickly, and this is an animation of this network that we traced out with electron microscopy. So very briefly, I just want to go through, zoom way out, and the types of experiments that are going on in my lab and the range of scales. So first, all of our experiments now are in the behaving mouse. Most of our behaviors are very simple, just running in a paradigm protocol that David Tank developed and was very fruitfully applied by Chris Neal and Mike Stryker to the visual system. And they found that when a mouse is running, it's standing on a styrofoam ball, and this is in our lab, the mouse begins to run. But you see its head is fixed, and they found that neurons in the visual cortex respond up to 3 times more robustly, more strongly, when the animal is running. So now let's zoom in from this behavior, simple behavior, to zoom in and rotate the animal and look down at the top of the mouse head. And what we're doing and other groups are doing is putting a cranial window on and a plate or a head post that holds the head still. And the observatory is through that little cranial window that Mark Anderman and colleagues in my lab developed. What we're really looking at is the cortex, and this is a very small diagram of the sort that Leah Krubitzer showed yesterday, but let's zoom in some more. Take the same diagram of the mouse areas, and this is from the work of Wang and Burkhalter. Wang is now at the Allen Institute, a figure made by Ma and Burkhalter. And these are all of the cortical areas in the mouse. And at the posterior pole, you see V1 and higher visual cortical areas. And now let's go to the analogy of the observatories. So it's a very nice analogy. And what is an observatory? A canonical observatory, of course, is a telescope. And in modern astronomy, there's all sorts of wonderful telescopes and wonderful surveys. One of the great ones is the Sloan Digital Sky Survey, which takes a quarter of the sky. And they took millions of images and made just a landmark survey of the stars and galaxies and and quasars and everything else in that quarter of the sky. So that's a wide field survey. But just as importantly, and sort of capture the public eye much more, is narrow field ones, where the Hubble telescope in particular looks for parts of the sky where there are very few nearby stars, and looking back really to the beginning of time, and all those wonderful pictures of hundreds of galaxies together is from the Hubble Deep Field or the Hubble Ultra Deep Field, where they can look back close to the beginning of time in the first billion years of the universe and see deeply and deep back in time. So it's not the worst analogy for what we want to do, that the wide field is at brain scale, and at the scale of the cranial window that we make, 5 millimeters is roughly a quarter of the visible cortex on the surface of the brain, and we are observing the night sky, or sorry, the southern hemisphere, the visual system, and that window can be shown here. That's an actual window, and then a couple of targets. This is beautiful work that I don't have time to tell you about by Mark Anderman, Aaron Kurland, and colleagues, Lindsay Glickfeld and Dimitri Rumis, looking at higher visual areas, thinking about issues of dorsal and ventral streams, thinking also about issues of projection patterns, all done with genetically encoded calcium indicators injected into these various areas. I won't tell you about that. What I will tell you about is the narrow field, the deep view, and just going back and forth, you might not see the little square that I'm bringing up. That is roughly 300 microns. I just threw it on PowerPoint. And now let's zoom in by another factor of 10. And to the image that I showed you at the beginning of the talk of 100 neurons in the mouse visual cortex. And now we're to the middle part of the talk, posing the questions. Again, large-scale physiology, and really, I often have talks titled Toward Complete Functional and Structural Imaging of a Cortical Circuit. Not claiming to have complete understanding, of course not, complete analysis of the data, no, but at least complete imaging where we can see all of the things that we want to see in the data set. First, I've already told you about large-scale physiology, but wouldn't it be nice to zoom in by another factor of 100? So we've gone from wide field imaging with 50 micron spatial scale, not quite pixels, but spatial scale of 50 microns. Now I'm about to show you roughly 5 nanometer resolution. So we've gone to a factor of 10,000 in spatial scale of imaging. Wouldn't it be great to zoom in to those two neurons that do completely different things? And these are actually a real zoom in. This is not PowerPoint. It is PowerPoint, but it's actually real. Two neurons that are right next to each other in vivo that we studied with electron microscopy at 4 nanometer resolution. And why do we need that resolution? And why do we need electron microscopy? Electron microscopy gives us the resolution, but the staining techniques allows us to see, without specifically labeling each neuron, every axon, dendrite, and synapse. So this is the modern generation of the visual physiology that we're doing now. The stimulus, Vincent Bonin in the lab, who now has taken a faculty job in Belgium. Wei Li, who did the electron microscopy along with Davi Bach, but now is also doing the physiology, and Lindsay Glickfeld. So a very rich stimulus, very rich response. On the bottom, you see it looks like fluorescence, but it's actually our electron microscopic data set. And the point of showing this is, let's do it again, is to just bring up the idea of slice anatomy, that we're doing very large-scale electron microscopy. But what it's giving us is a slice in a computer, where the same way a slice in a dish can allow people to patch neurons and probe connections, this slice in a computer allows us to trace through the neuropil and look for connections. So now I'll speed up a little bit, but the middle part of the talk is actually quite short, posing the question, the backdrop for those of you who aren't doing cortical neuroscience every day, cortical neurons, of course, come in two types, excitatory and inhibitory. You've heard of already from Nat Heinz, but we'll hear more from Han-Kui Zhang, and I guess you heard from Sasha Nelson yesterday. Of course, there are many, many other types. But just excitatory, inhibitory, excitatory neurons are the most numerous. And I'm showing cell bodies, dendrites, axons. And just to say that we can see all of these things for all the neurons in our data set, including synapses. Why am I really showing you this? Is that excitatory neurons are most numerous, and they're sparsely interconnected. Ballpark 10% of nearby neurons of the same type are interconnected. And then the question is, what is the structure of that sparsely interconnected network? And that's my question. That's really the question that many of us are interested in, and it's a big question. 1 minute of doing a computer analogy, that a computer too has some very basic elements and or not, perhaps XOR. And we know that, but no one would begin to say that's what a computer is, and we understand it by knowing that. We could look at very simple statistics, like I might guess that 20% of ORs are followed by NOT, so in fact 20% of ORs are NORs. But no one would, it's absurd, and that's why I'm doing this. No one would say that the statistics of connectivity at that level will tell us anything about computation. And for computation, we need motifs. And what's a motif? Here are some very simple motifs in computer science. Binary addition. A binary addition is order, roughly 10-ish AND gates put together in a very, very precise way that gives you 1 bit of addition with carrying both input and output, and you can string together 8 of those, and you get an 8-bit adder. I promise you, in logic circuits of computers, or in a GPU, there are millions of these things, and there are a smaller number of them added together in these larger motifs. That's how to understand the computer. So the question is, what are motifs in brain circuits, and how do they relate to the computation being performed? So that's where we come to what do we know about cortical circuits. 5 years ago, I think many people would argue that a cortical circuit is a network of sparsely interconnected neurons, and that's it, and that it's random with respect to that, and in fact, random networks can compute a lot of things. But of course, I don't believe that, and I think much of the field is not believing that. From a few slice studies from, Sasha Nelson was involved with really one of the very first slice study of this nature in work that he did in collaboration with Mitya Chklovsky, who analyzed their beautiful slice experiments. But there's work coming from Henry Markram and others that are saying that the networks are non-random, but we still don't know that the non-randomness, what the non-randomness might mean. And I believe that one can study that non-randomness in the context of what the neurons actually do. So are there cell assemblies? Probably. What do they do? We don't know yet, but one possibility is that neurons that do the same things are interconnected, and that might be a motif that has a function, and those neurons might be neurons that do different receptor fields, or more simply, they could be neurons that fire together. And any Hebbian rule will tell you this is how the brain is wired up, but there's no data in the cortex, in the actual adult cortex, that says this is true. So what did we do? More background, but I've told you this already. I just am showing, re-emphasizing the point that in rodent visual cortex, and we started doing this 6-7 years ago in the rat, finding that it's a really nice playground to ask questions like this, because in rodent visual cortex, neurons that do different things are mixed together. So the network appears random in terms of the placement of the components, but then what is the substructure of that? And started 6 years ago. We have a number of sort of opinion pieces about it. But then we are asking this question, last of the middle part of the talk, if you have functional imaging and then wiring diagrams, what regularities might we find? And just to head off a question, Of course, every brain is different, but the rules and the motifs might be the same. And so I've sort of embodied one model that neurons that have the same function might be interconnected well. There's a beautiful study from Tom Mercer-Schlugel's group in London that gave the first nice answer to this question, and it's likely to be a yes, but there are many forms of this question that we'll all be working on. What we addressed now in the last part of the talk was a simpler question, and that was a question posed by the thesis work of Aaron Kurlan in my lab, along with Mark Anderman, where they found that inhibitory neurons of multiple subclasses have little or no orientation selectivity. That this built on work from Tsumoto's lab in Japan, from Mike Stryker's lab again, and others. But there's a controversial consensus coming out that many interneurons are unoriented, and that would pose the model that these triangles, pyramidal cells that do different things, should converge on inhibitory cells that don't really care about that thing. But let's try to prove it anatomically by doing this experiment. So I've shown you this picture 5 times already, but now let's start about the anatomy. That little rectangle is the part of the functionally imaged brain that we, meaning Davi Bak, just an amazing graduate student who now has his own group at Janelia, and Wei-Chung Lee, a postdoc who continues this work in my lab, this came out in Nature 5 months ago or so. They drilled in on this one physiological experiment. They started with the physiology performed by Aaron Kurlan and Mark Anderman, and this is now in vivo data, the 3-dimensional structure at micron resolution of the brain in vivo. They took the brain, perfused the animal, took the brain, cut it in an animation that doesn't work on the screen, but they cut it physically along this plane where we cut the in vivo data computationally, so we can see the brain rotated sideways where we have the peel surface of the cortex. We see blood vessels in red. That's very important for aligning the 2 data sets, but I won't be able to tell you that part of the story. The calcium indicator, which is green, and most importantly, the plane of the brain where we had physiology from on the order of 10 cells. So we have the function of 10 cells. Now let's look at the network. How do we look at the network? We do electron microscopy, and this is the work of Davi Bak and Wei-Chung Lee and a large team. So this is step 2, taking the same brain and taking pictures. This is one picture, but as you'll see, it's a pretty extraordinary picture, that it's the same thing that you saw in vivo with EM. Cortical surface, the kind of white things are cell bodies, the very white things are blood vessels. In this data set, we have 1,500 cells, so it's very large scale compared to most electron microscopy. I wish Terry's talk would have come first. He would have seen that, but you'll remember to put Terry's talk right now. How many pixels is that, Christoph asks. Let's see. Sorry to be sarcastic. How many pixels is that? It's a lot of pixels. We're a great team. I love this. And this, this is sort of the show-off phase, but it's important to say why it's so big. We needed hundreds of microns across to encompass the arbors of axons and dendrites, but we also needed nanometer resolution, 4 nanometer resolution, to see the axons, dendrites, and synapses. Boy, I'll be 3 minutes over, Christoph, no more. You asked the question. So each one of these pictures is 9 gigapixels, 9 billion, 10 billion pixels, and we have 1,100 of these pictures, and that's 10 terabytes of data. We had 3 million 11 megapixel images stitched together, so ending up with 10 million megapixels of data. Again, that's cool in some sense, but we need it to have both the breadth and the resolution. High throughput imaging, lots of post-processing. I won't go through that. This is the data set just rendered so you can get a sense of it, that we have thousands of cells and blood vessels and such, still rendering the EM data set. And here are the red and the green cells that you saw earlier, rendered from this very large data set. But here really was where the action is. These are EM data sets. And this shows the three-dimensionality of it. There's a cut face where you can see on this face here, on the right face, the nucleus of two pyramidal cells, the beginning of the apical dendrite of two pyramidal cells. But it's nice to see that there's a third dimension as well. But now let's really zoom in for the action. I'm glad that Paul Allen just walked in, because I love his big screen here. Let's zoom in some more. But now let's zoom into native resolution. And this is why I like the big screen. This is native resolution, pixel for pixel, native resolution. And I was just sitting there looking. This is probably about 1 yard. And so it's 6 yards across. This is 1/100th linear of the data. So that's roughly 600 yards. And let's make it 700 yards. And that's exactly if this is just a random sampling of an image that is 100 acres, actually, at this scale. So it's a very big image, and we have 1,000 of them. So that's why it's big data, so we can see all these things. So, the cell bodies are easy to see. If you need to sort of learn to see EMs, but I'm telling you, that's a cell body. This is the important part. These circles are a bunch of axons cut in cross-section. And by hand, and by eye, you can see these quite well and trace them in 3 dimensions. But those are less than 100 nanometers, or roughly 100 nanometers. There's a synapse, and that's very important to see. We, meaning Davi Bak, Wei-Chung Lee, and Hyun Kim, segmented all this data by hand. Another story. The data are online. I believe very much, and certainly the Allen Institute believes very much in putting all the data online. So you can actually see the entire data set served up by both Mark Ellisman at UCSD and also our colleagues at Hopkins, openconnectomeproject.org. And you can browse through the data and even mark it up online. An enormous amount of work was done, yielding this movie where we have first the physiologically identified cells. They traced out the axons and dendrites of these cells. So these are the physiologically identified cells. We know what these cells did in the living brain, and that's the important part. We could trace out their axons, picked out of 1,000 other cells. We picked out these 10 axons, traced them, jumped the synapse, found more than 100, probably 200 postsynaptic targets. And that's the unique thing, that the resolution and the dense staining allows us to crawl this network and reconstruct the important to us 0.01% of the network. But it's just enough of this huge data set to answer a question. Do inhibitory cells get random input from excitatory cells? And the answer is yes. And I'll run through the last few slides. Here's a notional example of 2 cells that we know what they did in vivo, converging onto an inhibitory target. To take a note from my Harvard colleague who I just met yesterday, read the paper, and you can see exactly what was done. But this is an example of 2 axons traced to a common inhibitory target. And on the right is their physiology. That's the nice thing, to go back and forth, anatomy, physiology. This is the network. The colors are the function of the pyramidal cells. The other important color is cyan, the light blue. The inhibitory targets in this mess of a network, the inhibitory targets are those little cyan circles or squares. And here's an example looking at the whole network. Let me jump through this and go to the network. This is just the part of the network where we had 2 inputs from these cells that converged onto common targets. And we had enough of these that we had certain examples like this, which is exactly what was predicted from the physiology. Here are 4 excitatory inputs onto a common inhibitory target and 4 distinct colors. So that's just an existence proof that this kind of random convergence can happen. We have many more of these, some of them slightly less random, some of them that appear to be the same, but you'd expect as many similar as different if it were totally random. Final panel is that it's consistent with this model. Final real panel of the talk is that in the first iteration of this type of experiment, where we're trying to connect function and connectivity, we were able to address a simple question with a simple answer, that the excitatory to inhibitory connections appear random with respect to the function. They appear more like gain control than embodying the actual computation. I believe, and I think many people believe, and probably increasingly, that the excitatory to excitatory connections are the scaffolding or the structure of cortical computations. And in the next experiment that we're running now, but certainly the work that we hope to do here, that we will look at much more detailed physiology and much larger networks and try to see, again, the relationship between function and connectivity at a much larger scale. Most important slide, everyone who's done this, the calcium imaging was started a decade ago by Kenichi Oki in my lab and other colleagues, all of whom have gone on to start their own labs. I've told you the work of Aaron Kurland and Mark Anderman in particular, who started the mouse imaging, and I mentioned the work of Vincent Bonin and Lindsay Gluckfeld in particular. Very large team for electron microscopy. First and foremost, Davi Bach, who started the project, the whole thesis project, this whole thing, joined very soon by Wei-Chung Lee, who continues the work in the lab. It was absolutely necessary to have our collaborators at the Pittsburgh Supercomputing Center, who took the 3 or 4 million individual pictures and stitched them together into a three-dimensional aligned whole that allowed us to begin the tracing. And last, and critically important, was this all was done at the Center for Brain Science. All of the EM was done. I'm at Harvard Medical School. The Center for Brain Science at Harvard College has a project that Jeff Lichtman started and funded, or got the funding for, where I did all of this EM, and I owe a great debt of gratitude and collegiality to Jeff, where we had our various projects for the EM, and of course Kenny Blum and Josh Sains, the executive director and the director of the Center for Brain Science. So, thank you.